Good evening, my dears. We'll be starting our zoology discussion. The first question from zoology part. Start from question number 114. DNA polymorphism, that is difference at the molecular level, that forms the basis of both genetic mapping and DNA fingerprinting. It is based upon DNA polymorphism that we are marking the genes in a chromosome as well as DNA fingerprinting is done. Okay, which one of the following statement is not true regarding gel electrophoresis technique? Question number 124. Gel electrophoresis is used for separating molecules of DNA which are all fragmented using an appropriate restriction enzyme. So, the presence of DNA can be visualized by staining with ethidium bromide, that is correct. Presence of chromogenic substrate gives blue color. This is not correct because this is a technique for identifying a transformant from a non-transformant recombinant one. Okay, so when you are using an alternate method, when you are using a technique where you are using PUC, this technique will be easier to find out or to distinguish a recombinant transformant from a non-recombinant transformant. It is not applicable to this one. We see them as bright orange colored one, and process of extraction of separated DNA stands from gel. That is, we cut out that piece, that gel, and transfer it to a suitable solvent, that is called elution. So all are correct except for the second one. Okay. Next, given below are two statements. One is labeled as assertion. See, this type of questions are very, very important because you have to think correctly and correlate these statements. Polymerase chain reaction is used in DNA amplification. Ampicillin resistant gene is used as a selectable marker to check transformation. Correct. See, both are correct. If both are correct, how you arrive at the correct option? Polymerase chain reaction is used in DNA amplification because then ampicillin resistant gene is used. See, there is no connection between these two. Statements are all correct, but the statements are not related. So, we can give only one. Both A and R are correct statements, but R is not the correct explanation of A. It is not the correct explanation. Both statements are correct, but not the correct explanation. That is why we take the option 1, which is not a method of ex situ conservation. What is ex situ conservation? Out of the native, original place. That is the meaning of ex situ. In situ means in their original natural habitat. Okay. National parks are created for protecting plants and animals of a particular area. So, these are all created in their original place, in their native. It is not an ex situ, it is an in situ. Exoskeleton of arthropods. A very simple question. They are all made up of chitin. Chitinous exoskeleton. Chitinous exoskeleton. They are all having, they are characterized by chitin. What is chitin? N-acetyl glucosamine, a polymer of N-acetyl glucosamine. Next question is, again from biotechnology. If a geneticist use the blind approach, see, approach is blind. We are not using whether it is a expressed one or non-expressed one. We have to understand the genome entirely. Then we use this technique, blind approach, for sequencing the whole genome of an organism. That is followed by assignment of function to different segments. That is called sequence annotation. Then by assignment of function to different segments, the methodology adopted by him is called sequence annotation. Transposomes. It is mobile gene segments or DNA segments. Transposomes can be used during which of the following? It is used in gene silencing. Whenever a foreign RNA enters and forms a double-stranded RNA, or when a jumping gene, that is the meaning of transposomes. Transposomes are simply jumping genes. They jump from one position to another position. When they are all jumping, they will be producing a double-stranded RNA. In such conditions, gene silencing, which is also called RNAi, is operating. In which of the following palindromic sequence of DNA which can be cut easily by a particular restriction enzyme? A palindromic sequence is one which should be read in the same direction, same in both strands. See, if you are reading in 5' to 3' end, in the complementary strand also, you should read it in the same way and you should get the same sequences. So, GAATTC, it is from 5 to 3. So, 5 to 3, GAATTC. Everybody knows that this is the sequence of EcoR1. Which of the following will accelerate phosphorus cycle? Remember, phosphorus cycle is a sedimentary cycle. So, it is by the weathering of rocks that they will be coming back to the ecosystem or made available to the ecosystem. So, they will be accelerated by the phosphorus cycle. Sedimentary cycles are all accelerated by this weathering of rocks, because earth crust is the reservoir, okay, that will be releasing this material to the ecosystem. While explaining the interspecific interaction of a population, plus sign is assigned for beneficial interaction, negative sign for a detrimental, harmful, and 0 for neutral interaction. Which of the following interaction can be assigned for one species plus and another one minus? Okay. See, amensalism. In amensalism, one is always negatively affected and the other one is not at all getting any benefit. In commensalism, only one is getting benefit, the other is not at all affected. In competition, both are negatively affected. In predation, prey-predator relationship, it is the prey who is negatively affected, and the predator is getting its food. So, one is getting the benefit, the other is getting harm. Okay, so that is the correct option. The asexual reproductive structure associated penicillium. Penicillin belongs to ascomycetes. Ascomycetes are reproducing by conidia formation exogenously. That is characteristic of ascomycetes. They reproduce asexually by conidia spore formation. Again, we are having another assertion reason type question. Here we are having osteoporosis is characterized by decreased bone mass and increased chance of fracture. Common cause of osteoporosis is increased levels of, actually it is decreased levels of estrogen. Okay, it is given as such in our books. So please understand that A is correct and R is not correct. Only if you are all having both assertion and reason correct, we may go to the option whether R is a correct explanation or not. Here A is correct, R is wrong. So the option is 2. In gene therapy of adenosine deaminase, we are having a classical example for gene therapy. The patient requires periodic infusion of genetically engineered lymphocytes. The patient's lymphocytes were taken and genetically engineered lymphocytes were again introduced into periodically. Why? It is because that genetically engineered lymphocytes are not immortal cells. Anna, see, we are having the lymphocytes taken out, but lymphocytes are living only for a very short period. They will be living only for a short period. So, after that, they become dead. Even if you are all providing genetically engineered lymphocytes, they will be living only for a short period. That's why we have to repeatedly do it. So, genetically engineered lymphocytes are not immortal, they are all mortal. So, that should be the correct one. Again, in situ conservation. What is in situ? In their original place, we are all protecting all the plant, animal, species, so that future can have them protected and conserved in their original habitats. So, in situ conservation refers to both plant and animal protection in the original form. So, whole ecosystem is protected. So, the option goes to 4. Which of the following function is not performed by secretion of salivary glands? Salivary glands contain salivary amylase, thiolin that digest complex polysaccharide like starch into disaccharides. Lubrication of oral cavity, the mucin, the mucus is helping in lubrication. It is not digesting disaccharides. Polysaccharides are converted into maltose, disaccharide. Then it cannot digest disaccharides into further glucose or monosaccharides. So, digestion of disaccharides is not done. Control bacteria population in the mouth. It is containing lysozyme. So, because of the lysozyme and also because of the presence of certain chemicals, bacterial growth is inhibited, suppressed. Okay. So, digestion of polysaccharides is not taking place. Nitrogenous waste is excreted in the form of pellet or paste. See, ammonia, urea and uric acid are the 3 forms of nitrogenous waste materials, of which ammonia cannot be discharged, urea cannot be discharged in the form of pellets. So, the question is, which are all excreting in the form of uric acid? Clear? Salamandra, amphibians. Amphibians are ureotelic. Hippocampus, fish, monotelic. Pavocstatus, the bird, the national bird, Pavocstatus. So, it is excreting in the form of birds and reptiles are typical, insects are the typical uricotelic, ornithorhynchus mammal, ureotelic. So, the answer is Pavocstatus. Again, multiple statement questions. We are having a lot of such multiple statement questions. mycoplasma can pass through less than 1 micron filter size. That statement is correct. They are the smallest living forms, mycoplasma. And mycoplasma can also change their shape. So they can pass through. Even if the size is very small, it can pass through pores which are all less than 1 micron. Mycoplasma are bacteria without cell wall. That's why they are capable of changing the shape. That is why they are called jokers of microbiology. Okay. So statement 1 is correct. Statement 2 is incorrect. So statement 1 is correct. Statement 2 is incorrect. That is the option. Question number 160. Which of the following statement are true for, that is applicable for spermatogenesis and not good for or not holding for, which is applicable for spermatogenesis but not applicable to oogenesis? It results in the formation of haploid gametes. That's a correct statement. Differentiation of gametes occur after the completion of meiosis. See, differentiation of gametes occur after the completion of meiosis. Why it is not applicable to oogenesis? See, in male gamete, differentiation of gamete take place. Spermatid will be undergoing the second stage. That is called spermatoleiosis or spermiogenesis. Then only it becomes a gamete. Okay. Whereas in oogenesis, immediately after meiosis, it is ready for fertilization. So, differentiation of gamete occur after completion of meiosis. Is applicable only for meiosis, only for male gametogenesis, because only in them you will be having the second stage, that is differentiation, spermatoleiosis or spermiogenesis. Meiosis occurs continuously in a mitotically dividing stem cell population. Why it is so? It is a continuous process. Whereas in female, it is broken. There are two stages at which it is arrested. It is not a continuous one. Meiosis occurs continuously. After prophase 1, it is arrested, stage 1. And the arrest lasts for 12 to 13 years. This will be taking place at about the time of birth, and the remaining process of development will be taking place only after attaining puberty. So, it is not a continuous one. In the case of males, spermatogenesis is a continuous process. So, it is applicable to male but not applicable to female gametogenesis. It is controlled by the luteinizing hormone. In both cases, it is correct. It is initiated at puberty. Major events of both male and female gametogenesis are taking place after puberty. But see, in the case of males, we are having the events taking place in puberty. Whereas in females, even before birth of a female child, the process of gametogenesis will be taking place. Initial stages will be completed even before this one. Okay, so you will be having, it is initiated at puberty. It is applicable to the male, not to female, because in female it is starting even before a girl child is born. So it is initiated in puberty only for male, but not for female. So we will be having B, C and E occurring. Fatty acids and glycerol cannot be absorbed into the blood. Actually, fatty acid is immiscible with the blood, Glycerol is miscible with water, so can be absorbed. But in our book it is given fatty acid and glycerol cannot be absorbed into the blood directly. Specialized lymphocytic capillaries called lacteals carry chylomicrons into the lymphatic vessel and ultimately into the blood. So, in the light of the above statement, choose the most appropriate one. We are having two statements. These two statements are all correct. Fatty acid is indirectly entering into the blood. For that, we make use of the lymphatic system. Only through the lymphatic system it is entering into the blood. So, both statements are correct. So, we have to mark 4 as the correct one. The digestive tract has additional chambers like crop and gizzard. They are present in birds. So, the question is, which are all the birds, the group which is including only birds? Pavo, Pavocristatus, peacock, Citacula crameri, parrot, and corvus, crow. So, that is the correct. Before, amphibia, catla, fish, crocodile, reptile, okay, and chameleon, reptile. So, we don't have all the other groups entirely of birds. Only 3 is the correct one. The release of sperm into the seminiferous tubule is called spermiation. Correct statement. Spermiogenesis is the process of formation of sperm from spermatogonia. No, this is not correct. It is the formation of sperm from spermatids. From spermatid. So here, statement 1 is correct, 2 is incorrect. So the option goes to 2. Which of the following is not a connective tissue? Adipose tissue is a connective tissue, cartilage is a connective tissue, neuroglia. Neuroglia are special type of cells which are non-conducting, non-activatable cells of the nervous system. So, that is the correct option. Blood is a connective tissue, fluid connective tissue. Glia cells or neuroglia cells and neurons are the two types of cells of nervous tissue. Statement 1. Autoimmune disease is a condition where body defense mechanism recognize its own cell as foreign. Normally, our immune system will be recognizing our own body cells and differentiate it from non-body cells, self from non-self. Here, the statement is a correct one because where in autoimmune disorder, a condition where body defense mechanism is attacking our own body cells. So, defense mechanism recognize our own cells as foreign body and attacks them. Rheumatoid arthritis is a condition where body does not attack cells. Rheumatoid arthritis is a typical example for autoimmune. Okay, so we are having a condition where statement 1 is correct, but statement 2 is incorrect. Lippis loop. What is that Lippis loop? It is a non-medicated IUD. Lippis loop is a non-medicated IUD. Which of the following is not a function of conducting part of respiratory system? Conducting part is having 3 important functions. They are all humidifying the air, keeping the temperature increased, bringing it to the body temperature. The air we are all inhaling is brought to the body temperature. It is providing surface for diffusion. No. Through them, no exchange of gases are possible. It cleans. Because of the mucus and hair, like cilia, the dust particles and other foreign materials are cleared off. So, These are the three functions performed by the conducting system, whereas this function is performed by the exchange part. Which of the following is correct match of disease and its symptoms? Tetany. Tetany is because of low calcium level, not high calcium level. Myasthenia gravis. Myasthenia gravis is an autoimmune disease. Muscular dystrophy is a genetical disease. Arthritis, it is the inflammation of joints. So the correct one is option 4. Under normal physiological condition on human being, every 100 ml of oxygenated blood can deliver 5 ml of oxygen to the tissue. Coagulum is formed of network of threads called thrombin. No, it is formed by fibrin threads. Coagulum is formed by fibrin threads. Okay. Fleet is the graveyard of erythrocytes. Correct. So, statement 1 is wrong. Statement 2 is correct. Statement 1 is incorrect, but statement 2 is correct. So, option goes to 3. At which stage of oogenesis the process is initiated? In the case of a female, gametogenesis, that is oogenesis, in a female is taking place or initiated even before she is born. During the embryogenic stage itself, all the oogonia are produced even at the time of her birth. So, at a very early embryonic development stage itself. Tegmina. What is tegmina? It is also called elytron. It is a covering wing. This covering wing derives from mesothorax. So the answer goes to 1. The metathorax is bearing, only meso and metathorax are bearing wings. The metathorax is having membranous wing. It is having membranous wing. So only 1 is the correct option. Which of the following is present between adjacent bones in the vertebral column? In a vertebral column, the adjacent vertebrae are connected together by intervertebral discs. These intervertebral discs are made up of cartilages. Okay. Actually, there are fibrous cartilages. Intercalated discs are seen in the cardiac muscles. This is intervertebral disc. Intervertebral disc. Again, statement questions. Restricted endonuclease recognizes specific sequence to cut DNA known as palindromic nucleotide sequence. Restricted endonuclease cut the DNA strand a little away from the center. That is why you are having staggered end cuttings. Okay. So, we are having the enzyme cutting here and cutting here, not in the middle of that palindromic sequence, but a bit away from the middle. So, both are correct. Statement 1 and statement 2 are correct. Option goes to 4. detritivores. Detritivores are eating, just like earthworm that is given in our book. Break down detritus into smaller particles. This process is called fragmentation. Humification is a process where humus is formed in the soil. Decomposition is the overall process which consists of fragmentation, humification, catabolism, leaching, etc. etc. Catabolism is a enzymatic process. Okay. And here it is large sized particles convert into small sized particles. That is fragmentation. Given below statements. All vertebrates are chordates. But all chordates are not vertebrates. That's a correct statement. Why? Because notochord is replaced by a vertebrate column. We are having some chordates which are having no vertebrate column. That is why all chordates are not vertebrates. All chordates are not vertebrates, but all vertebrates are chordate because they have a notochord either in the early stage or throughout their life cycle. So that statement is correct. Notochord is replaced by a vertebrate column in adult vertebrates. That is why all chordates are not vertebrates, but all vertebrates are chordates. So that is the correct explanation. Both A and R are correct, and R is the correct explanation. All vertebrates are chordates because vertebrates will be having the notochord in the early stages, and all chordates are not vertebrates. There are some chordates, cephalochordata and urochordata. For all chordates, they have a notochord but not a vertebrate column. So they are not vertebrates. So that's the correct explanation. In the taxonomic categories, which hierarchical arrangement is ascending order? Ascending order means from the smallest to the largest. That is correct in case of animals. See, it should be starting from the smallest to largest. But there is no such order given. So we can take only this one as the correct one. Actually, it is in a descending order. Kingdom is the largest, species is the smallest. But the order is species, genus, family, order, class, phylum, kingdom. Okay. In others, species, genus, family, class, phylum, order. It is not in the correct order. Species, genus, family, phylum. It is not in the correct ascending order. Species, genus, order, family. Not correct. So the only one in the hierarchy which can be in the order can be this one. But this is actually in a descending order. Largest first and smallest later. Our taxonomic hierarchy is ascending, starting from species and lasting in kingdom. Regarding synapses, what are synapses? Synapses are junctions, either between a neuron and a muscle or between two neurons. Okay. Electrical current can flow directly from one neuron into another across an electrical synapse. That's correct, because the two neurons are in close contact, or the synaptic cleft is so small that electrical impulse can jump from presynaptic to postsynaptic one. Chemical synapses use neurotransmitters. Why they have to use neurotransmitters? Because the synaptic cleft is very large. So that the signal cannot jump from presynaptic to postsynaptic. Impulse transmission across a chemical synapse is always slower. Why? Because once a synapse signal reaches a synapse, the synaptic vesicle has to be activated. It has to fuse with the presynaptic membrane, release its neurotransmitter. Neurotransmitter has to diffuse across the synaptic cleft and fall on the receptors. Then only an action potential will be generated. So it is very slow. Impulse transmission across a chemical synapse is very slow. The membrane of presynaptic and postsynaptic neurons are in close proximity in an electrical synapse. Correct. So, which is incorrect? 3. Now, as I always tell you, whenever such a matching question is there, you have to use your intelligence. Bronchioles. Oh, what are these bronchioles? Bronchioles are having ciliated epithelium. Okay. So, one should be for immediately you can arrive at the answer. Goblet cells, they produce mucus, they are glandular. Tendons are dense regular. And adipose is a loose connective tissue. So, to save time, you should use that technique. Okay. We are having bronchiole, which is having the only correct option 4. 4 is the correct option. So, even if you don't go to other options, or even if you don't go for B, C, D and others, you can immediately arrive at the answer. Human insulin is given below. Which statement are correct about genetically engineered insulin? See, this is a question that may be a bit confusing for you. Why? Correct about genetically engineered. Pro-hormone insulin contain. See, our genetically engineered insulin does not contain any such C-peptide. A-peptide and B-peptide chains of insulin were produced separately in E. coli. That part is correct. Extracted and combined by creating disulfide bond between them. That's correct. Insulin used for treating diabetes was extracted from cattle and pigs earlier to genetic engineering. Before we invented humanin, this was the technique. But this is regarding genetically engineered one. So, in genetically engineered one, we are not extracting the insulin from cattle and pigs. Pro-hormone insulin need to be processed for converting into. In genetic engineering, there is no such maturation because they are all produced in 2 separate chain forms and then fused together. Okay. Some patients develop allergic reactions to foreign insulin. See, if you are taking insulin from cattle and pigs, then it will be causing some allergic responses. But for genetically engineered humanin, there is no such a problem. So only B is correct. So the option goes to 1. With respect to acquired immunity. Acquired immunity is one that we get during our lifetime. Anamnestic response is elicited on subsequent encounter with the same pathogen. First encounter is primary response and the second encounter by the same pathogen causes. Okay, so that's a correct statement. Anamnestic response is due to the memory of the first. That's why it is very stronger and faster. Acquired immunity is non-specific. No, it is specific against the particular pathogen only. So, it is not correct. Primary response is produced when our body encounters a pathogen for the first time. That also is correct. So, the wrong statement is 3. Again, we are having match the following question type. Whenever you are having that match the following type, we have to use the technique. For example, diaphragm. Diaphragm is a technique, contraceptive methodology that cover cervix, blocking the entry of sperms. So A is for, we are having two options. So please be careful. Okay. Contraceptive pill. Contraceptive pills are inhibiting ovulation and implantation. So B is one. Again, both of them are going neck by neck. IUD, intrauterine device, increase the phagocyte of the sperm. This is correct. Only when we reach the third level or third set, we will be arriving at the correct answer. Lactation amenorrhea, absence of menstrual cycle and ovulation following parturition. Okay, so the option goes to 1. Correct statement. Bacteria are exclusively heterotrophic organisms. No, we are having autotrophic, chemoautotrophic, photoautotrophic. They are exclusively heterotrophic is not a correct one. Fly molds are saprotrophic organisms classified under Protista. They are protestant fungi. Mycoplasma have DNA, ribosomes and cell wall. They are bacteria without cell wall. Cyanobacteria, blue-green algae, are group of autotrophic organisms classified under the kingdom Monera. That's a correct one. There are actually monerans, and formerly they were all included under plantae algae. That's why the old name blue-green algae is still retained by them. Okay. Which one of the following statement is correct? The tricuspid and bicuspid valves open due to the pressure exerted by the simultaneous contraction of the atria. See, tricuspid and bicuspid valves they are all opening by the pressure exerted by the simultaneous contraction of the atria. These valves are also opened. Okay, you will be having the valves open when the ventricles are coming back to the original size diastole, so that 70% of the blood from atria enter into the ventricle. Blood moves freely from atrium to ventricle during joy and diastole. Atrium will be first coming to the diastolic state, so atrial filling take place. Now ventricle also come back to the diastole stage, and during that the pressure inside the ventricles are decreasing and 70% of the blood is coming. So that is the correct one. Increased ventricular pressure cause closing of the semilunar valves. Now it will cause closing of the dicuspid and bicuspid valves. Atrioventricular node generate the action potential to stimulate atrial contraction. It is the SA node, the pacemaker that generate an action potential to stimulate atrial contraction. So only correct one is the second one. Which of the following is not a desirable feature of a cloning vector. See, a cloning vector should have some qualities. What are these qualities? A cloning vector should have one, presence of a marker gene that help you to selectively clone the transformant recombinant one, presence of a single restriction enzyme for a particular enzyme, because more than one restricted site for the same enzyme will cleave the vector into so many pieces and complicate the entire process. Presence of two or more recognition site, you don't want it, because if a vector is having more than one cloning site, cutting site, it will be cut into so many pieces and it will complicate the entire process. So a vector should have only one cutting site. Presence of origin of replication. Without ORI, it will not automatically replicate. So it should have ORI. So third one is the undesirable character present in it. Parathyroid hormone. This parathyroid hormone, what is its action? It is a hypercalcemic hormone. Stimulate the process of bone resorption. Resorption means demineralization, removing the calcium deposit from the bone. Decrease calcium level in the blood. No, it is hypercalcemic. Resorption of calcium by renal tubules. Calcium will be absorbed back from the renal tubules. Decrease the absorption of calcium from digested. No, it is increasing the absorption of calcium. Increases metabolism of carbohydrate. It is having no such role. So here, which of the following are not the effect? Not the effect. So B, D, and E. So option goes to 1. Again, we are having biological molecules. Match the following. Glycogen. What's a glycogen? It's a stored product. Animals are storing this one as the excess food material in the form of stored food, in the form of glycogen. So it's a stored product for what is globulin, immunoglobulin? It is antibody. What are all steroids? See, steroids are hormones. They are also acting as hormones. And thrombin, it's a biocatalyst. Thrombin is essential for blood clotting, converting fibrinogen to fibrin. So it is 2. So 4, 3, 1, 2. The option goes to this one. Okay. You have done all the questions perfectly. Nothing is out of syllabus and there are no much problems with the questions. Okay. And only thing is that questions may be lengthy. I have told you that. You have to read every statement using your fingers up to the last word. If you have used it, you should have scored almost every question correctly. And out of the 45 questions, as we always say, more questions will be from first year. 27 questions are from first year and the remaining 18 are from second year topics. That is a proportion. And biotechnology is one of the chapters from where you are getting a lot of questions. Biotechnology 1, chapter 1, 5. And among the first year topics, we are having two chapters from where four questions are there: biological classification and animal kingdom. These are all as per our expectations, and all chapters are covered, and there will be no problem for you. I think so. And with that, I conclude my discussion. Thank you all. Thank you.